Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is an event that's been going on for almost 3 years now, since the beginning of the COVID-19 pandemic in many countries. And we're very excited today to talk about a potential solution to a big problem that people are having in the field. And that is, how do you actually label your extracellular vesicles after they've been produced in order to be able to track them, to understand where they're going, how cells are interacting with them. And one of the solutions that people have used for many years is lipid dyes, and they can be very valuable with the right controls. However, there are some challenges that we're going to hear about today, and that means that we need some new solutions. So today's event is co-hosted by SNEV, the Student Network on EVs. And so we have Natalie Turner here today from SNEV to co-moderate the event. Actually, she's going to be doing all the moderating. I'm going to be lazy today and hand it over to her. So Natalie, thank you so much for joining. And I'd like to ask you to maybe say a few words about SNEV and then introduce our speaker today.
For sure. So thanks for having me. Obviously, it's my pleasure to welcome you also to this week's EV Club in conjunction with SNEV, which is the Student Network on Extracellular Vesicles. So SNEV is a virtual network run by students who are interested in the EV field. Our mission is to connect EV research students internationally, and we are the first virtual platform for students to discuss, share knowledge, network, and establish collaboration with other students and ECRs in the EV field. And we offer this through a series of virtual events mostly. So as Ken mentioned, I'm Natalie Turner. I'm a PhD student in the Mitchell Group at Queensland University of Technology in Brisbane, and I'm also a SNEV committee member. So today's SNEV EV Club is with Sarah Cox-Vazquez, who is currently undertaking a postdoc at Singapore National University. She'll be presenting her group's work titled Water-soluble extracellular vesicle probes based on conjugated oligoelectrolytes. And this was recently published in Science Advances. So Sarah, thanks so much for joining us today. I'll let you introduce yourself as well and get started with the presentation.
Thank you so much, Natalie, and thank you so much to ICEV and SNEV for inviting me to give this talk. I'm very excited. Just a little bit of background on me. I'm American. I got my PhD from the University of Michigan in 2019, and then I moved to Singapore right at the start of the pandemic in January 2020 to start my postdoctoral fellowship in the lab of Guillermo Bazan. So I'm just going to go ahead and share my screen. Hopefully you can all see that. Someone could please let me know? That'd be great.
Yeah, perfect. Thanks, Sarah.
Great. Thanks so much. So we're really excited to talk to you today about, a new type of dye platform and how it can be useful for work in EVs. So like I mentioned, I'm from NUS. That's where I'm doing my postdoctoral fellowship at the Institute for Functional Intelligent Materials in the lab of Professor Guillermo Bazan. Just a disclaimer, we have decided to start to commercialize these dyes under the brand name Aquarella, but today will be more of a scientific, academic focused type of presentation, so we can all learn a little bit more about what makes conjugated oligoelectrolytes, or COEs, useful for EV research. So I don't think I need to give the big how EVs are produced and all of that background. So I'd like to talk today more about how to specifically probe EVs and how many of you and myself have been looking at, how to track EVs, how to look at them on different methods like flow cytometry, for example. So here in this image, we have the cross-section of an EV. And if we were to look at this, we see a few identifying features. So we have our surface marker proteins, some nucleic acid or internal protein cargo, and all of these have different ways that we can probe them. For example, dye-conjugated antibodies on the surface or fluorescent engineered proteins are very popular, as well as perstaining with DNA or RNA intercalating dyes for the lumen of the EVs, as well as bioconjugation dyes like CFSE, for example. So all of these dyes are quite useful depending on the question that you're trying to ask for these multiple parts of EVs. But as we all know, that EVs are quite heterogeneous in these different types of markers that we're seeing. But one thing that is quite unanimous across all EVs are the lipid bilayer. And so the lipid bilayer is quite a popular target, I would say, for interrogating EVs. And people are kind of using a more classical type of membrane dye, which looks something like this. So here is the chemical structure of PKH26, but other popular dyes like DIR or DID, for example, are quite similar in their overall chemical structure. So if we were to think about the lipid bilayer, we have, two portions of lipids that allow it to form this bilayer. We have the hydrophilic head groups as well as its hydrophobic tails. And the classical membrane dye chemistry very much mirrors this about lipids. And so that they have these aliphatic tails that help them anchor into the bilayer, but they also have a dye portion on the head group. So up at the top is what would actually make the dye portion of these types of dyes. But because it's quite a mimetic of just a single individual lipid, it does come with a few different issues. So they're actually not water soluble, which leads to the formation of micelles and these nanoaggregates, which tend to give these false positives in the buffer control. And so these are quite, they make these form their own micelles, which are about 100 nanometers in size, which are very much in the same range of the EVs that we're interested in looking at. So here we have a few different methods that you can commonly see these false positives in just your controls. So here we had just PKH26 and buffer, and on methods like flow cytometry, looking at the violet side scatter versus the fluorescence intensity for PKH26, we can see a very defined population of a fluorescent particle, which is just the PKH26 in the buffer on its own. And we can also see that very nicely demonstrated by DLS, which is a more bulk solution type of method to characterize the nanoparticles. And we can actually even see these particle formation by naked eye using the Tyndall effect, where simply in a dark room using a handheld laser pointer in a solution of PKH26 and PBS, if we were to shine the light of the laser pointer into the vial, we actually see a lot of scattering happening just from a handheld laser and looking at our eye. So we know that this is quite a problem, because if we were to mix these aggregates in with our EVs, they would be roughly the same size and be very difficult to purify or isolate with very simple washing methods. So this is what drove us to, really pursue this work. So I would say in the Bazan lab, we actually have been working on conjugated oligoelectrolytes for quite some time now. We've been working on making these different types of molecules for a variety of applications, including OLEDs as well as for antibiotics, but we had never applied them more as optical probes. And so in early January 2023, we were able to publish this paper in Science Advances. And so that's what we're going to be talking more about today. So a conjugated oligoelectrolyte, or COE, as what we'll call them for the rest of this talk, are a water-soluble small molecule that actually mimics the domains of lipid bilayers. So instead of mimicking, the overall architecture of a single lipid, now we're mimicking the entirety of the lipid bilayer. So we have the hydrophobic core of the COE, which is this conjugated portion here, as well as some hydrophilic tails with some charged end groups. And these COE molecules are, because we're a synthetic chemistry group, we're able to very nicely kind of mix and match them and play around with different properties that we're looking at, kind of like Lego blocks, for example. So it's actually the center portion of the COE or the dye that actually gives it its overall color. And if we want to be able to change the color of the COE that we're looking at, we can simply change out a few different parts of the molecule, kind of like Lego blocks, and that can help us to change the color. So in our paper that was published earlier this year, we presented a new palette of different types of conjugated oligoelectrolytes, COEs, that have a variety of different excitation and emission in order to, facilitate these more as dyes for multiplexing and for other downstream applications. So we were able to show a palette of 8 different COE molecules that have quite a range of different excitation and emission properties. We showed all the way from, UV excitation that we see down here in the purple that can be excited at a 365 laser, all the way to a near IR2 emission that can be excited using an 808 laser. So we were very excited about this work, excited how, this might be able to help EV researchers, being able to mix and match and use them in their own assays. And so here in this table, we just have some photophysical properties of the dyes, as well as looking at their cytotoxicity. So we're very excited to say that they're very low in cytotoxicity to A549 cells, because of their high level of biocompatibility. So if we were to take a look at how, next slide, there we go. If we were to take a look at how this might actually look once it's intercalated into an EV, for example, in comparison to a classic dye, we see that because of the overall architecture of the COE, it's able to intercalate itself fully into the lipid bilayer. So it's able to span from the inner to the outer leaflet. This is due to its overall molecular structure that mimics the overall domain of the bilayer instead of just mimicking a part of it. And because of its chemical design and its overall chemical length, it actually very much matches the thickness of the lipid bilayer, which helps it to be quite biocompatible and non-toxic. So one of the nice things about COEs is that when they're actually in solution on their own, they're not emissive. So when they're just in water or buffer, they are actually dark. So they don't have any emission on their own. But when you mix them with a lipid bilayer containing particle like an exosome, we can actually see them, even by naked eye detection, we see them become quite emissive and light up. So here in this small video that we're showing, on the left-hand side, we have just the COEs in water. And then on the right-hand side, we have the COEs that are embedded in exosomes. And so again, we're just holding it under a handheld UV light to be able to see, this emission happening in real time. So I think in the very beginning of my talk, I talked about the motivation of wanting to use the COEs as probes for EVs. And one of the big reasons was because COEs, we know that they're water soluble and they don't form their own lipid nanoparticles. So here's just a schematic explaining a little bit more in depth why we think that COEs would be a good probe for EVs, is because if we compared them directly to a carbocyanine dye like PKH26, the COEs are water soluble and hence don't form micelles. So we've talked a lot about how this could be, the theory of the application, but let's take a look more in depth at the data. So using flow cytometry, we were able to show as our first method that the COEs don't give any false positive signals if we were to compare them to PKH26. So using two different instruments that are quite commonly used for EVs, both the Cytoflex, the Beckman-Coulter Cytoflex, as well as the Nano FCM Nano Analyzer. On the left-hand side, we show COE 600, which is just 1 micromolar in water, and we don't see any of these large particle formations like we see with PKH26. So this is good, showing that these aren't forming their nanoparticles and can be easily applied with these types of methods like flow cytometry, for example. And to demonstrate this a little bit further, because we are, a chemistry group, we wanted to show this also by absorbance and fluorescence. So here we're using an ultrafiltration, so an Amicon ultrafiltration tube in order to demonstrate the water solubility. So on the left-hand side, we have COE 600. So we can see in the top of the Amicon, it's quite dark in color, a purple color. And on the right-hand side, we have DIR. So we can see the blue solution there. So if we were to put this into a centrifuge and centrifuge it for about an hour, we see that the COE can very easily pass through the ultrafiltration filter. So this is a 100 kilodalton cutoff ultrafiltration filter, which has a pore size of about 11 nanometers. So we can see that the COE is able to very nicely pass through, which we can also see demonstrated by its absorbance. So before filtration in the blue line, we have the COE 600 control. So we can see a very nice absorbance spectra from just the dye on its own. And after ultrafiltration, because the dye is able to pass through the 11 nanometer pore filter, we can see that we have the same spectrum before and after. However, for the DIR dye, we see a very blue solution in the top of the ultrafiltration filter. And then after spinning through the centrifuge, it actually goes clear, indicating that somehow these larger particles that the dye is forming is becoming trapped within the filter and unable to pass through, which indicates that it is forming some kind of nanoparticle that is in water soluble and getting trapped in the filter. So a few more properties about the COEs is that they're only emissive in the presence of the lipid bilayer. So here we're showing in blue, we have the COE 490 with SUVs just in PBS, so a lipid bilayer mimetic. And then we also have in black, you can see at the bottom, the COE 490 in PBS. If we were to mix it with FBS, so very common for cell culture, but highly rich in proteins, and less so in lipid bilayer particles, we don't see any emission coming from the COE in this highly protein solution. And importantly, we've also shown here in dynamic light scattering that in the presence of SUVs, again, like in the top panel with 10 micromolar of the COE, we don't see any change in the size of the liposomes or SUVs, indicating that it is not damaging them in any way. We also showed this by cryo-TEM. So we wanted to be able to see if the lipid bilayer was remaining intact. So in the top portion, we have our liposomes, just the control, so no dye has been added. And then on the right side, we have added the same equivalent as COE 490 as in the past experiment, so 10 micromolar. And we don't see any disruption or damage of the lipid bilayer, showing that they're still intact. And we saw the same thing for A549 EVs. For them, we see the control has a very nice defined lipid bilayer, and we have some density coming from the inner portion of the EV, indicating some kind of cargo that is within there. And then when we add the COE, we see the same types of structures. No damage is happening to the EVs, so they're quite compatible with both of these systems. So now I'd like to get into a little bit more of how we can use these COE dyes for applications in EVs and some more downstream applications like flow cytometry. So due to their water solubility, you don't need any type of wash or purification steps. So if you have your isolated EV sample, you can simply mix with the COE dye at room temperature at 37 for just 30 minutes to 1 hour, and then the dye will spontaneously intercalate into your EVs. And then you don't need to do any type of washing or purification. So here we're showing some flow cytometry plots from the Beckman-Coulter Cytoflex. So in the left-hand side, we have just our EV control. So these were some PC3 exosome standards that are commercially available from Abcam that have been purified and lyophilized after ultracentrifugation. So on the left-hand side of the screen, we have just our EVs versus the violet side scatter versus the fluorescence intensity. So just in PBS, we can see that we still have some background noise, some events that is coming from PBS alone, just from the electronic noise that comes from using this instrument at a very high sensitivity. And then in the bottom row, we just have the PBS with these three different dyes that we're showing. So the COE 430, the COE 490, and the COE 600. So these three dyes are optimally excited at 3 different channels. So we can detect them at the UV 450 channel, as well as the violet 525 channel or the blue 690 channel. So if we go to the top row of these plots, we have over on the left-hand side, we just have the exosomes by violet side scatter. And we can see that we have a lot more events, a lot more density happening due to these particles going through, passing in front of the laser. But because we don't have any type of dye, it's a lot more difficult to get any kind of relevant or useful information from this. So if we were to mix the dye in the protocol that I showed in the beginning, we can see that after the dye intercalates, we actually see that the EVs themselves become much more emissive. So for the Aqua 430, we have 49% positive particles, the 490 up to 76%, and then the COE 600, 36.8%. So we were very excited about how we can use this and what this indicates for downstream applications for EV detection. And one of the questions that we got asked quite often is, okay, so after you label these particles, what happens to the dye? Does it leach out? Can it cross-interact between other particles? So for that, we use some small unilamellar vesicles stained with equal concentrations of either COE 490 or COE 600. So we stained the liposomes with those two different dyes, and then we mixed them at different ratios and allowed them to sit at room temperature for either 1 hour or for 24 hours. So here we have from the Cytoflex 2 fluorescence intensity plots, either the COE 490 or the COE 600. And we can see that even after mixing for 1 hour, we don't have any type of dye transfer or any swarm effect that's happening in the dual positive region, so quarter 2. And even after mixing for 24 hours, this is just on the bench at room temperature, not stored in the fridge, just covered under a piece of tin foil to protect it from light. We don't see any type of interparticle dye exchange or fusing of these vesicles yet. That would indicate, a single particle carrying both of these dyes. But we performed the same experiment using the traditional classic dyes, so DIO and DID, which have the lipid tails with a dye head group at the top. And just for mixing at 1 hour, we saw lots of interparticle dye exchange between the two of them, indicating that even though the dye has originally gone into one particle, it's quite easily transferred between the other two particles. So for EVs, and we want to be able to get out as much information from a single particle as possible. So antibody co-staining is a very popular method for analyzing EVs on flow cytometry. And so we want to be able to see if the COE dyes are also compatible with that. So we do know that the dyes don't light up when they bind to proteins, and they only light up when they bind to a lipid bilayer. So we want to be able to test whether the fact that it was biocompatible to be able to use 2 types of dyes, a protein-based dye with our COE dye in one. So we were able to come up with a very simple staining protocol with no wash or purification needed. So we were able to incubate the PC3 exosome standards, again from Abcam, with some commercially available PE stained antibodies from BioLegend. So we mix them for 1 hour at room temperature, and then we can analyze those via flow cytometry. And then we can add our COE dye to the same mixture and then use that same sample again on flow. So here we just have the exosome that is only stained with CD9 antibodies labeled with PE. So on the x-axis, we're showing CD9 positive, CD63, and CD81, as well as a cocktail of all 3. So while staining for antibodies is very useful, it's difficult to tell if we're only seeing a portion of the EVs or if we're actually seeing the full population when we're doing this co-staining with these 3 different markers. So staining with the COE dye allowed us to see how much of the population we're actually able to pull out of the noise from there. So here we have our co-stain sample that are stained with CD9, again on the x-axis, and COE 490 on the y-axis. So we can see that this is on the BDA1 small particle detector machine, that we're able to see 55% positive with our COE 490 dye. And then if we were to look at the CD9, 63, 81, and then the cocktail, we see the same distribution of positive labeled exosomes with them. So if we were just to look at the antibody alone over on this bar graph, for CD9, we had about 17% positive, followed by 6% positive for CD63, 4% for CD81, and then all of the cocktail was 29% positive particles. And then when we stained the same samples using COE490, we see the same distribution of antibody positive events, but now we also have 55% that are positive for COE490. So this would allow for a better picture of the overall EV in a sample, as well as a breakdown for how many labels are on each individual EV, as well as the total population. So this would help to see a little bit more of the EVs out of the noise. And excitingly, we didn't see any type of artifact induced by mixing the COE with the antibody, showing the high compatibility of the COE dye with different markers. And so for downstream applications like in-cell tracking, for example, of EVs, it's quite important that even though the dye is water soluble, it's important that it can be removed quite easily. So we were able to come up with a staining protocol that allowed for quite simple removal. Earlier, we showed how easily the COE dye can actually be removed with an Amicon. So in this experiment, we wanted to be able to compare how this would enable some tracking using imaging flow cytometry. So if you were to simply stain the EVs with the dye, and then you can remove the excess dye and retain only the stained EVs using the ultrafiltration. So you would be able to put in your stained EVs in the Amicon, spin to remove any excess dye, and then being able to recover your just stained EVs for further downstream applications. So to begin, we wanted to see just the dye-only controls. So previously in the earlier slides, we did show you how the DID dye was able to get stuck in that Amicon filter and was unable to be removed, and we weren't able to recover its signal by absorbance. And here, if we were to perform the same experiment just with the dye-only controls without any EVs bound, all of the COE dye was able to be washed through. Actually, one second, let me just explain that this is imaging flow cytometry. So here, instead of using the laser just to detect fluorescence, it actually uses a camera to be able to detect the individual cells. So it's quite useful for being able to see how dyes are distributed amongst a cell, which can be very useful and powerful for monitoring EV uptake into cells. So here we have, the bright field, the fluorescence image, and then the fluorescence overlay of both of them. And then down here, we have more of our traditional flow cytometry plots where we have the size or bright field area of the cell versus its individual fluorescence intensity. So either the COE490 or the DID dye. So performing these dye-only controls, we were able to observe that we didn't see any signal coming from the COE490. So nothing was retained within the filter of just the dye in buffer. However, with the DID dye, we see lots of these particles that are actually stuck in the filter, which is a nice practical application of our former experiments, showing that the DID dyes are forming these particles and they're getting stuck in the filter, which are not easily removed. Which is why more traditionally you use protocols like ultracentrifugation or size exclusion to be able to get rid of these bigger aggregates. So in a more functional type of assay, we wanted to see how labeling EVs with these two different dyes would differ in this same type of experiment, removing the excess dye with the Amicon filter. So we compared the COE490 labeled EVs versus the DID labeled EVs. And we wanted to monitor over time how the EVs were being uptake into cell culture media. So here we monitored at 2, 4, and 8 hours using the COE490 labeled EVs. And over time, we can see that as the EVs are incubated in the cell culture media with the cells, we see more and more fluorescence and more individual bright spots that are coming into the cells and into the fluorescence image that are nicely distributed amongst the cell. However, for the DID labeled EVs, we see much more of these kind of big aggregates that are happening, and there's much less precision and much less more defined spots that we're able to resolve using these DID labeled EVs. And this is due to the insolubility of the DID dye forming these nanoparticles. And for that reason, it's more difficult to be able to have more precision and more more precision in being able to correctly identify whether this was actually an EV spot or if this was a dye aggregate spot. And the nice thing about flow cytometry, it's actually able to quantify the overall uptake of the EVs using its mean spot count. So being able to actually see the whole cell and see how the fluorescence is distributed within the actual cell is very powerful to see how many of these EV spots are actually being internalized. So on plot A, we just are looking at the overall number of labeled cells. So after 8 hours of incubation with the COE labeled EVs, we saw that almost 100% of the cells had internalized some kind of EV. And then after 8 hours, we were able to count the overall number of spots, and we saw about 16 total spots plus or minus 15 within the EV. That had cells that had taken up a COE labeled EV. So recently, very excitingly at NUS, we just acquired the Nano FCM Nano Analyzer. And we're quite excited about this because we think it's going to be able to enhance sensitivity of some of our dyes. So in a few slides back, I showed you our COE 600 that was labeled with some PC3 EVs. And we didn't see a very high percentage of total positive events. However, with the same dye and the same concentration of EVs, we were actually able to see a much higher positive ratio of labeled events. And this is due to the fact that we think that the Nano Analyzer is an instrument that is designed for the nanoparticles in mind. So it has a much slower flow rate, so there's a much longer integration time of the labeled particle that is passing in front of the laser, leading to enhanced sensitivity. And what was also very exciting for us when we were looking at the Nano Analyzer is that it was able to help us to visualize the size of some RBC EVs that were with and without labeled with the dye. So these RBC EVs were kindly donated to us by our collaborator. And what was very exciting for us is that even on the previous, the Cytoflex, we only ever saw about 30% positive population. Now we're able to see 100% of the labeling of the EVs on this instrument. And it can very help us nicely see that, again, the COE isn't actually changing the size of the EVs once it's actually intercalated into the dye, into the EV. So with that, I have a lot of people to thank for this work. So I'd like to thank Prof. Guy Bazan for, being the advisor and overseeing this work and helping to really drive it forward, as well as the other members of the Bazan Lab that helped to make this possible. I'd also like to thank the flow unit staff who was so helpful to me in learning how to do flow cytometry and apply it to EVs, as well as the other members of the company, Aquarella Private Limited, that's based here in Singapore, Basie Lim, as well as the incubator program here at NUS that has really helped us to get off the ground. So with that, I'm happy to take any questions. Thank you so much.
Great. Thank you so much, Sarah. That was a really great presentation. We have a question from Geordie. So Geordie, do you want to unmute yourself and ask your question, please? He just said, hey there, really nice work. So first of all, do you treat the Amicon filters in any way so you don't lose the EVs? Because I've been working with them and some other colleagues, and we have noticed that the EV concentration is extremely reduced. And secondly, how do you think that the hydrophilic tail from your dye can cross the lipid bilayer?
Okay, great question. So I'll start with the Amicon question first. So no, the answer is no, I don't treat the Amicon with any type of detergent. I've seen people use like a non-ionic surfactant to treat the Amicon before use, but that's not necessarily necessary in my case. I found that I was able to get good recovery. The key that I've noticed is the recovery spin is very important, and you really have to make sure to wash the stuck EVs that are stuck onto the filter very vigorously with whatever buffer you're going to resuspend it in. With that, I was able to get a higher recovery of my EVs for these types of experiments. But I do think that no matter what, you're going to lose some of your EVs to the filter. You're not going to get 100% recovery. But with good washing and the recovery spin, you're able to get quite a lot back.
Okay, great. So I'll just add one extra question on before you move to the next one. So was that the only method of recovery or washing that you tried with the COEs, or did you, because a lot of with the lipophilic dyes, we find that people are doing ultracentrifugation to wash. Was this, how many methods did you try before you settled on this one?
Before I settled on this one, for this paper, this was the method we knew we wanted to use to be able to demonstrate the water solubility. We have used ultracentrifugation and size exclusion to be able to remove the excess dye, and both of those work quite well. We just didn't add these to the paper, but I can tell you they also do work. They are just more tedious and more time consuming to use those types of methods for this removal. So we think that, you know, the water solubility and the easy processability is quite nice for these kind of specific applications.
Okay, excellent. And then, yeah, so that second part, oh, sorry, is that Jordy?
No, yes, I couldn't
Yes,
unmute me, but now
Okay.
I'm there. Thank you for your answer. And well, the second question that I had was, how do you think that the hydrophilic tail that has your dye can cross the lipid bilayer? Because if there's the hydrophobic part of the lipids, how do you think that it can cross it?
Yeah, great question. So if we come back to the structure of the COEs, they do have an intrinsic positive charge. So the first type of interaction, and we've done some MD simulations as well as some other type of more imaging that will be published in the future. But we do know that it's driven more by the electrostatic interactions first. So the electrostatic of the positive charge to the negative charge particles of the EVs or other types of lipid particles. So driven first by the charge interactions, then the intercalation of this really hydrophobic portion of the dye to actually get in within the lipid bilayer.
Mm-hmm. Thank you so much.
Sure.
Okay, so Sarah, sorry, I have another question. So I wondered, the EVs that you used in the paper were like cell-derived, and some were liposomes, correct?
Right, right.
So I wondered how, if you've tested the dyes with EVs from complex biological fluids like plasma or milk, and if so, how did the dyes perform, and what would, you know, obviously contain a lot of other lipid particles, not just EVs?
Right. So we do have some collaborators that we worked with that have done a lot of testing on milk EVs, for example. They're currently using them for tracking for delivery as well as for tracking for flow cytometry. So we do know that they are applicable for those types of EVs. In more complex fluids like plasma, for example, that do have lots of other contaminating particles like lipoproteins. That's something that we're very actively looking at right now within our own lab, is how, you know, with these kind of more lipid monolayer type particles that would be the lipoproteins, what kind of selectivity can we see between the COEs and the more monolayer particles like the lipoproteins versus the bilayer particles like the EVs. Yeah, we do have some other collaborators that have used them for purified plasma samples, for example, so that they've isolated the EVs from plasma and then have done some antibody panels, for example, to look at different antibody markers. So yeah, there's lots of work happening on a variety of EV types.
Yeah, that's wonderful. Thank you so much. Okay, so I think the next question is Larry. Larry, did you want to unmute yourself and ask your question, please?
Yeah, hi, Sarah. Beautiful presentation. Thank you. And thank you for the samples that you sent us.
Yeah.
Can you tell me, we found with some of the other stains that we use that vesicles from one cell type will stain much more efficiently than vesicles from another cell type. Have you tried comparing, because I think most of what you presented was a single cell type as the vesicle, where the vesicles were originating from. Have you looked at that at all?
For the multiple cell types, so we personally have looked at HEK derived EVs, some RBC derived EVs, MCF7, and the PC3 EVs that we've shown. So they're mostly cell culture derived, except for the RBC EVs, and we do see pretty consistent staining amongst all of them. We've also done a few more internal studies looking at different liposomes of a variety of different compositions, for example, varying cholesterol and varying, the lipid head group. We did see pretty consistent labeling amongst all of them. Maybe the reason that they might have different efficiencies of staining is perhaps you could have particles that have a really high amount of protein that is embedded, and then they have less room for a lipid bilayer dye to go in. That could be one reason that you could see differences amongst EV staining. But for us personally, with the different types of EVs from cell culture or milk, for example, we do see pretty consistent labeling.
Great, thanks.
Yeah.
Okay, great. So Isabel, you're the next question. Can you unmute yourself, please?
Yeah, hello, really nice talk, thank you. Just wondering if you try to disrupt the stain EVs with detergent as Triton. If so, did you lose the signal?
Yes, the signal is obliterated after detergents from Triton or SDS.
Okay, thank you.
Perfect. Okay, so there's no more questions in the chat box, so I have another question, Sarah. So with regard to the lyophilized EVs that you used in the paper, I wondered if you compared the performance of the dye between the lyophilized versus the freshly isolated. I think I have a personal interest in changes to the EV membrane following lyophilization. I just wondered if you compared that at all, or if you plan to compare that if you haven't.
Great question. So the lyophilized versions, we get them from Abcan, so they're commercially available. They come in a lyophilized powder. We've also done internal, ultracentrifugation, as well as size exclusion to try and purify the EVs. We have a figure in the supplementary information of this paper looking at if we can still stain them isolated from different methods by flow cytometry. And we did see that we still get a very nice brightly stained population for both of those types of samples in comparison to the lyophilized sample.
Okay, great, awesome. Okay, Nadia, your question is next, please.
So questions. So first, I remember you shown in the first slides, you measured pKa sizes in buffer on the DLS. Did you try to measure your dye sizes in PBS on NTA or DLS?
Yes, we did. I think I missed that slide in this talk, but because the dyes are water soluble, they don't form those nanoparticles. So let me just go ahead and jump through. Let me see if I can find. Yeah, because they're not forming the nanoparticles, there's simply like no signal that comes from the DLS. It's just kind of a very blank, looks very similar to what PBS would look like. That is within our paper, and I'll send you the paper and I'll point out the exact figure to you so that you can see it.
Okay, thanks.
Yeah.
Did you try to compare photo bleaching time of your dye comparing to the other lipid dyes?
Yes, so we did a direct comparison between the COE versus DIR, for example, and we saw very equal photo bleaching times between the two samples.
Okay. I don't remember the slide, but there was figure from the Cytoflex. Maybe this one also could. Did you try? Did you make a calibration with some particles? If yes, which particles did you use?
Yeah, so they're pre-calibrated with a 100 nanometer and a 300 nanometer particle to be able to see the size distribution to be able to set the threshold correctly for the individual cytoflex. So they're 100 nanometer silica particle from, I think, Thermo. I'd have to go back and check where we bought the particles from, but yes, we do calibrate the instrument before we do the runs.
Okay, can I email you about this question? Just interested.
Yeah, sure. My email is on the very first slide. I'll just pop it back up on the screen. Just either Sarah at Aquarella or my NUS email is fine too.
Thank you.
Yeah, of course.
One more question. So I like the picture, how dilocated in the membrane. How did you know that this is exactly the same location as in reality?
So right now we are undergoing some studies using some polarized microscopy to see the orientation of the fluorophore within like a GUV, for example. So we have a very talented student in our group who's working on publishing that. And we can see that it is oriented parallel with the lipids within the bilayer, but we have not published that work yet. Previously, we've done some MD simulations to see how the dye actually goes in. So that's how we do know that it's driven by some electrostatic interactions first and then internalization. So I wouldn't say we have like exact structural information using like NMR or crystallography, but we do know from a variety of methods that this is the overall kind of picture of how they would be inserted within the bilayer.
Nice. All right, thank you so much.
Thank you so much for your questions.
Okay, does anyone else have any questions that they'd like to ask now? I think that's everything in the chat box for now. So Sarah, I think we might be wrapping up. Thanks again so much for doing the presentation today. It was so interesting, and obviously there's a lot of interest in getting samples from you, so I think you'll be a busy lady in the next few weeks. But yeah, so thanks again on behalf of SNEV and on behalf of the EV Club. Ken, did you want to say anything before we wrap up?
Only to reiterate the thanks. So Sarah, this has been wonderful. It's been really educational for me, and I think you can see the interest of the audience. And thanks to you, Natalie, and to SNEV for organizing this. So take care, everybody, and hope to see you again soon.
Thank you all so much. I really appreciate the invitation.